In our blood, we have three main cellular components. We have erythrocytes, the mature RBCs, red blood cells. We have platelets, also known as mature platelets, of course, thrombocytes, and we have leukocytes, the topic of our discussion today. Leukocytes, white blood cells. Red blood cells, as we know, are responsible for transporting oxygen. Platelets help in blood clotting and prevent excessive bleeding, right? And white blood cells, they are involved in defending our body against infections. Now, white blood cells or leukocytes. White blood cells, as the name suggests, white blood cells, the cells that are white. Leukocyte literally means leuko mean white and cyte means cells. So leukocyte literally means white cells. Now here is an important thing. After staining, right? Whenever we have to observe things, we stain them. After staining, when we observe these cells, white blood cells under microscope, they are not actually white, right? They may appear purple, blue, or have different shades depending upon the stain that we have used. The reason they are called white blood cells is because, unlike red blood cells, they do not contain hemoglobin, right? The protein that carry oxygen. They do not contain hemoglobin. Therefore, they appear to be colorless or white in their natural state, right? So in our body, in our natural state, if we are able to see white blood cells um, before staining, right, or without staining, then these cells will appear to be white or colorless. That's why we have named them leukocytes. Before we go further, I think it is important, and let's pause for a moment and think about something. Nothing on this planet actually have color on its own. For example, if I'm holding a green pen and I say it is green, but is the pen truly green? Not exactly. What is happening is a combination of light and our eyes. When light falls on the pen, most wavelengths are absorbed, but the wavelength corresponding to green is reflected. The reflected light enters our eyes, and our brain interpret it color as green. So the color is not the in the pen itself. It is the result of how light interacts with the object and how our eyes and brain process that information. Now imagine this pen placing on Mars with no human or without any visual system to observe it. The pen will still reflect certain wavelength of light, but there would be no green as an experienced color, because color is ultimately a perception created by the viewer, right? The same idea applies to blood cells. We call them white blood cells or leukocytes, but when we actually observe them under microscope after staining, because of course we have to stain them, right, they appear purple, blue, or sometimes nearly transparent, right? So that's why we are calling them white, because just like pen, they are not truly have a fixed color. In their natural state without staining, they are essentially colorless, but the term white simply distinguishes them from red blood cells, which appear red due to hemoglobin, right? The hemoglobin absorbs certain wavelength of light, and it reflect the wavelength of light that is corresponding to red color. Our eyes interpret it. So interestingly, if there is the thing that I want to tell you is that, interestingly, there is no real color, right? There is no real color. We are calling them white blood cells white, red blood cells red, because how we see them, right, how we interpret them. So now leukocytes, the topic of our discussion, are broadly classified into two main types. We have granular leukocytes and we have agranular leukocytes. Granular leukocytes, granular, granular, and agranular. Granular leukocytes are those white blood cells that contain visible granules in their cytoplasm. These granules are packed with enzymes and toxic substances that help destroy pathogen quickly. Right? When the pathogen enters our body, degranulation occurs. These granular leukocytes, they release their granules. These granules have enzyme and toxic substances and chemicals that will digest the pathogen, destroy the pathogen quickly. Because of this, granulocytes are mainly involved in body's immediate non-specific immune response, which is also called as innate immunity, right? The type of immunity that is not specific. For example, if we have two pathogen A and pathogen B, both have entered our body. So against these two pathogens, the response of our body will be same, right? Because it's non-specific. But because pathogen A is different and pathogen B is different, our third line of defense, that is specific immunity, right? That is involved in acquired immunity of our body, right? The term that we name them active immunity, passive immunity, right? There are four type of acquired immunities. So this immunity will respond specifically. Pathogen A, the response against pathogen A, the antibodies that will be produced against pathogen A will be different as compared to the pathogen, as compared to the antibodies that will be produced against pathogen B. That's specific immunity. Granulocytes are those white blood cells that are involved in immediate non-specific immune response. We have granulocytes, we have neutrophils, eosinophils, basophils, right? And their cytoplasm is filled with granules. If I talk about neutrophils, and you can see the historic picture of neutrophil on your screen. Neutrophils, they have different loops in their nucleus. They are polymorphonuclear. Why it help neutrophils in in immediate response, right, of our body? How it helped neutrophils to go, because whenever there is an infection, neutrophils are the first to arrive at the site of infection. Why, or how, we will talk about this in another video. But neutrophils, they have polymorphonuclear, they have 2 to 5 loops, and in their cytoplasm we can find few in number granules. We have eosinophils that are again polymorphonuclear. Their nucleus is bilobed, and their cytoplasm has granules. And because it has a lot of granules, then the cytoplasm appear red or pink in color, because these granules absorb the pink stain, right, and make the cytoplasm pinkish in color. Then we have basophils that are like least abundant white blood cells, leukocytes of our body. These basophils again are bilobed, and their cytoplasm are filled with granules. And there are a lot of granules. That cytoplasm is almost not visible because large number of granules. So the concept here that I want to tell you is that the white blood cells, the leukocytes, that are involved in non-innate, that are involved in the type of immunity in your body that will respond fast, non-innate, right? Fast, innate, and non-specific. They are classified. These white blood cells are classified into granulocytes. Why granulocytes? Because they have preformed toxic substances, preformed enzymes in their cytoplasm in the form of granules. Whenever pathogen will come, because they don't need to, the word that I'm using here, they don't need to study the pathogen. From studying the pathogen, I mean, previously I have given you the example of two pathogen, that pathogen A and pathogen B, that have entered our body. The response of our body against pathogen A in third line of defense will be different as compared to the response for pathogen B. Why? Because our body will study the pathogen A and pathogen B. Will study the antigens. Antigens means antibody generating substances. Will study the antigens that are present on pathogen A and pathogen B, and after studying the antigens, it will produce these a corresponding antibody, and this antibody will attach to their particular antigens, right, and it will detoxify them. There are multiple things that antibodies, there are multiple ways how antibodies show their response. But this is what I'm trying to tell you: the white blood cells that have to perform immediate, that have to perform non-specific response, our nature has classified them into granulocytes, right? They have already pre-formed substances in their granules. Whenever pathogen will come, they will immediately release the toxic substances. The degranulation will occur. These granules will contain enzyme or toxic substances, and they will help. These will help will to destroy the pathogen. On the other hand, agranulocytes, these don't have clearly visible granules under microscope, right? They have, or in other words, they have extremely few granules in their cells. These cells are mainly involved in specific or adaptive immunity. We, if I talk about granulocytes, then the word that you can use is Ben. You know, Benton cartoon. Benton. Basophils, eosinophils, and neutrophils. Basophils least abundant than eosinophils that are a little bit more abundant. They are in our total. If I talk about total differential leukocyte count, then eosinophils are 2 to 4%. Then we have neutrophils that are most abundant white blood cells of our body. We have 60 to 70% of all the white blood cells are neutrophils. Why neutrophils are the most abundant leukocytes of our body? We will discuss them in another video. Agranulocytes, these white blood cells are involved in specific adaptive immunity, right? And if I have to study the antigen, right, it will take time, right? So this takes more time to develop, but it is highly targeted, meaning to say pathogen A will, the response against pathogen A will be against pathogen A and not against any other pathogen. The agranulocytes includes, we have lymphocytes, and we have monocytes. Lymphocytes such as B cells, why we are calling them B cells? Because these cells were first discovered in bursa or fabrica, that's an organ in birds. And T cells, why we are calling them T cells? Because T cells are the lymphocytes that matured in thymus gland of our body. That's why we have named them T cells. So these cells are responsible for producing antibodies that will directly attack the infected cells, right? Monocyte can differentiate into macrophages and help in engulfing and preventing pathogen to other immune cells. So in summary, the presence of granulocytes and agranulocytes allows our immune system to respond in multiple ways: a rapid, immediate response, defense response that will be shown by granulocytes, and a slower but highly specific and targeted response that will be shown by our agranulocytes, right? So that's the main concept here, why we need two type of leukocytes in our body.